All right, everyone, we're going to go ahead and get started. Thank you so much for being here today. We have, of course, are very excited for today's speaker. Before we get into all of that, let me just do a couple of little announcements. My name is Cinnamon Moffett. I'm the research program manager here at Hatfield. And this is a hybrid event. So we have folks in the room and folks online. Folks that do this with me a lot know this little spiel, but I'll just do it again. For folks online, your cameras, mics, and screen shares have been disabled for this event, but we ask that you communicate with us through chat. We have a volunteer in the room. Roseanne is here with us. She's going to help make sure that you can hear and you can see. And if you have any technical issues, just let us know in the chat box. That's also the place you can put any questions for today's speaker. And Roseanne will read them out when we open up for questions so that both folks in the room and folks online and our speaker know what questions need to be answered. For folks in the room, again, if you just have a question at the end, if you raise your hand, you will need a mic to ask that question so folks online can hear you. You can either get this mic from me and I'll come bring it to you, or you can use the mic stand that's on the side of the room. I also am going to remind folks in the room, if you haven't, make sure you sign in on the sheet outside. That sheet does a couple things for us. It helps us justify with the university buying cookies and coffee. Sorry, folks online. There's cookies and coffee for those that join us here on site. But it also lets our speakers know how many people are participating in these outreach events so they can integrate that into any of their grants or responsibilities that way. So if you can help us accurately get those numbers, that would be great. Okay, a couple of quick announcements about upcoming events. Wanted to let people know or remind them that we've got a Science on Tap event March 20th. Megan Wingrove from the College of Engineering with colleagues from CEOS are going to be talking about making measurements at the face of an actively calving tidewater glacier. So that is going to be a really interesting talk with some amazing video. So if you want to join us, that is April 20th. It'll be in this room and online starting at 6 p.m. Also wanted to promote next week's seminar on Thursday is going to be Caitlin Magel, who is with the Puget Sound Institute at the University of Washington, Tacoma. We'll be talking about the declines of eelgrass in Coos Bay. So that will be a really interesting talk as well. So if you are interested in any of the events here at Hatfield, you can go to our events page on our website, scroll down and you'll get a calendar of events, and that'll tell you all the information you need to be able to join us either online or in the room. But the reason you're all here is for today's speaker. I cannot be Lee, but I'm going to try to introduce Dawn. And so forgive me for not being Lee. Lee is not able to be here with us today, but she was hoping to introduce Dawn. Dawn Barlow is a postdoctoral scholar with the Geospatial Ecology of Marine Megafauna, or GEMS lab, with the Marine Mammal Institute here at Hatfield. Dawn's research interests are in ecology and conservation of marine mammals. In particular, she studies what drives, when and where they are found, and how their distribution overlaps with human activities. She uses tools such as distribution modeling and spatial statistics, bioacoustics, and oceanography. Dawn first joined the Marine Mammal Institute in the summer of 2016, and we just reminded each other that's exactly when I joined, so we've been here the same amount of time. After receiving her bachelor's in biology and a BA in environmental policy from Pitzer College, here at OSU, she completed both of her master's and her PhD in wildlife sciences, advised by Lee Torres. Her graduate studies focused on ecology of blue whales in New Zealand. And as a postdoc, Dawn has been applying her spatial ecology toolkit to the collaborative project, examining the distribution of cetaceans and seabirds in Oregon and the California waters. And so I'm going to hand this off to Dawn, and she's going to tell us what she's going to talk about, and go for it.
I think got the green light. I think that means we're live. Thanks, Cinnamon. Thanks everyone for coming out on this really beautiful day. I was going to say no fault to anyone for bailing when it's sunny like this, but thanks for coming anyway. Lovely to have everyone here. So if you've chatted with me in the past, 7 years, I guess, you've probably heard me talk a lot about New Zealand blue whales. And today we're going to talk about something entirely different, much nearer to shore and much closer to home. Also smaller animals, but just about everything is smaller than a blue whale, I guess. So today, excited to share some work that we've been working on throughout my postdoc here in the Marine Mammal Institute. And a lot of this is pretty new recent work. So I'm excited for sort of the first major audience to be you Hatfield folks, and excited to gather feedback as we sort of enter the wrapping up phase of this study. Focused on gray whales and harbor porpoises in the nearshore waters of the California Current. Before I talk about any of the scientific results, I want to acknowledge that none of this is work that I did alone. So I want to acknowledge the wonderful co-authors on this presentation, Lee Torres here from OSU, Craig Strong of Crescent Coastal Research, and Barb Mueling, who's at Southwest Fisheries Science Center. And this work was supported by the Oregon Gray Whale License Plate Funds and contributed to by many folks who have really helped shape these ideas that we'll be talking about here. All right. So this will be no surprise to all of you, but anyone who's spent any time in the coastal waters of the Northern California Current or of the California Current here knows that it's characterized by a whole lot of wind. It's really windy. In fact, you don't even have to go on the water to know how windy it is here. And that's really important in this ecosystem. This is a wind-driven upwelling system where surface winds, sort of longshore surface winds, and the rotation of the earth lead to this net displacement of surface water offshore. That's replaced by deeper, colder, nutrient-rich water that's brought to the surface near shore into the photic zone, and that leads to this abundance of life. And these upwelling systems contribute disproportionately to fisheries catches and really have a high value both in terms of their ecology, economic value, and cultural value. So nearest to shore in the Northern California Current, the habitat is further shaped by sort of a complex mosaic of habitat features, including rocky reefs, sloping sandy bottom substrate, kelp forests, and shoreline topography like headlands and capes. And that makes for this really interesting and rich and complex and productive ecosystem and habitat right in our backyard here. So today we're going to focus on two different predators that rely on these nearshore waters, both marine mammals, with some sort of key differences in their life history and their biology. We're going to focus on gray whales, which migrate to these waters for feeding. They rely on this area for feeding on invertebrate prey, and harbor porpoises, which are here year-round. There are a number of different stocks that rely on these waters, and they feed a bit higher up trophically on forage fish and squid. So let's talk a little bit more about these species. Harbor porpoises, these images are from the stock assessments from the West Coast here, and there are a number of different stocks of harbor porpoises along our coast. In our study area that we're going to focus on today, it covers 3 different stocks of different sort of population abundances. But based on some recent genetic work, we know that these major biogeographic boundaries like Cape Blanco do contribute to shaping the population structure. And in fact, that's going to warrant some revision of these stock boundaries that's being discussed as we speak by the scientific review group for these stock assessments. Now, gray whales migrate from breeding grounds in Baja California, Mexico. Most of them migrate to feeding grounds up in the Bering and Chukchi Seas and comprise this eastern North Pacific stock. But there's a subgroup that shortens that migration and spends their summers feeding off of the coastal waters of the Northern California Current up to British Columbia. And that's a much smaller subgroup. And I'll just give a plug here that folks in the Marine Mammal Institute and the GEM Lab do a lot of really important research on these PCFG or Pacific Coast Feeding Group gray whales. So thanks to them for example, the beautiful imagery that you'll see today and this lovely animation by Lisa. And the ongoing work on these coastal gray whales here. And these are nearshore marine mammals inhabiting these nearshore waters, which means they overlap with nearshore human activity. So humans rely on these coastal regions for shipping and commerce, for fisheries, for industrial development, increasingly for the potential of renewable energy development. And so this this basically emphasizes the need for understanding the distribution and abundance and habitat use patterns of these species so that we can better understand where they're coming into conflict with human activities to sort of thoughtfully manage for conservation of this ecosystem. So our objectives here in this study were to evaluate the drivers of distribution patterns of these two different species and examine changes in their abundance over time for these two different coastal marine mammals. And the way we're able to do this is with a really remarkable data set motivated by studying marbled murrelets, a seabird species of conservation concern. And so since 1992, field teams have been conducting regular, standardized nearshore transects in these coastal waters. And these coastal waters are pretty tricky to navigate boats in. And so it has to be this small boat work going in and out of a lot of different ports up and down the coast. And so it's really pretty remarkable that throughout all of these years that they've been doing this work, studying marbled murrelets, they've also been recording other seabird species and the locations of marine mammals. Lucky for us. So how this goes in the field is as this little boat drives along a predetermined track line, there are two observers. One is looking on the starboard side, one on the port side. And so if they make an observation, they record a number of different things. Their location, the distance of that observation from the track line, the group size, so the number of animals they've observed, and also really importantly, the observation conditions. Because you can imagine if it's like really foggy or super windy, you're less likely to be able to pick up something small and fleeting like a little harbor porpoise. So over time, this has amassed a really incredible data set that we're fortunate to work with. What this heat map is showing is survey effort. So this big paneled figure here has year on the x-axis. The y-axis is latitude bins, and the darker boxes that are more black are areas with more survey effort. So they're filled in by number of kilometers of survey effort. And over, this 31-year data set, there's been more than 55,000 kilometers of survey effort. So we took this data set and implemented density surface models, which let me walk through what we mean by this, because it sort of underpins the rest of the results I'll tell you about. So as we said, these observers are going along a track line, they make an observation, and you can imagine, so they're looking for animals like this, but in reality, they're looking for something more like that. And you can imagine that under different conditions, you will be more or less likely to pick up something small like a harbor porpoise or a gray whale for that matter. And so it was important for us to account for our probability of detecting these animals. We do that by fitting a detection function to these distances so that we're able to estimate what's called the strip width. So that's how far we're able to estimate density under different survey conditions. And then we implement that into these distribution models. So density surface models are basically a special flavor of species distribution models. And I'll step through what we mean by that. So species distribution models are a great tool that allow you to take observed locations in space. For example, here, a track line with some critters sighted along it. And then you can take different environmental covariates that you're interested in, so depth, temperature, etc., and extract those at your observation locations. That allows you to put those into a model to understand the functional relationships between different predictors you're interested in and your response. And so in this plot, I'm showing it here as a schematic because we'll look at it with real data in just a minute. On the x-axis is your different environmental predictors of interest. Again, that could be depth, temperature, what have you. And on the y-axis is showing it says partial response, because that's the impact of any predictor on the occurrence of your species while taking the others into account. And what that allows you to do is to predict distribution patterns in space and time. Anytime you have data that your environmental data that you're interested in, you can make predictions of your species occurrence patterns, which is pretty cool. So what we did here is we accounted for the detection probability in these models. We used a number of different static and dynamic environmental variables that were available since 1992. So you can imagine that sort of limited our ability to use, say, the latest and greatest satellite products. We used GAMs, or generalized additive models, a favorite among ecologists because of their ability to fit nonlinear and complex relationships. And then we predicted the density of these different species we're interested in at a daily scale across our study area. So this is my main methods slide. If you have questions, I'm happy to talk about it at length, but for now, let's move on to the ecology we're interested in. So we had this hypothesis that gray whale distribution patterns reflect different features that influence nearshore retention, because that might elevate the productivity or the prey availability that they have access to in these nearshore habitats. So for example, in the case of coastal upwelling dynamics, we know that alongshore wind stress leads to this water movement offshore that's replaced by this deeper water through coastal upwelling. But when there's a relaxation in the winds, that offshore transport is reduced, and we get nearshore retention of nutrients and productivity that may enhance prey availability in these very coastal waters. So we hypothesized that there might be some sort of an optimal combination of upwelling events and relaxation events that are sort of the best case scenario or the sweet spot for gray whales feeding. There are other features that we think might be important, that we thought might be important, like the presence of rocky reefs, prominent capes that also shape these sort of recirculation patterns, and likewise areas where the shelf is wider, again, because of recirculation and retention of nutrients in the near shore. So let's take a look at the gray whale data. This is, again, a multi-panel colorful figure, but let's walk through it. This is showing the encounter rate, and that's the number of whales observed per kilometer of survey effort. You can think of it as catch per unit effort for fisheries. And it's being summarized in a few different ways. That multi-color situation there is showing year on the x-axis and latitude on the y-axis, and in each of those little boxes is the encounter rate, with the warmer colors being more whales encountered. And then across the top, it's encounter rate by year across all the latitudes, and then along the side, you have encounter rate by latitude across all the years. We could spend a long time looking at this, but I'm just going to pull out some interesting bits, which is the emergence of these sort of long-term stable hotspots in areas around Newport, Cape Blanco, and off the mouth of the Klamath River. And these seem to be stable across the decadal sort of timescales in our data. We don't see at least any real clear latitudinal shifts among years or over time. And so this is sort of what we can get at from looking at the raw data. So now let's go ahead and look at what came out of our distribution models that gave us these functional relationships with a whole range of different environmental predictors, things like depth and distance to the coast, distance to headlands, estuaries, etc. So here we go. So here are those functional relationships, and let's here the response curves are shown for. Let me back up. We did this by region. I'm actually going to go back to here and say we divided our study area up by different regions, and that's because there are some known really important sort of oceanographic and ecological characteristics of the California current that differ by region. And there are these sort of boundaries, biogeographic boundaries, we can call them, around major features like Cape Blanco and Cape Mendocino. And so because there might be region-specific habitat use patterns or prey preference patterns or distribution patterns, we modeled these regions separately. Okay, now we can look at these. So here again, we have the impact of each of these predictors on gray whale abundance is on the y-axis, and each predictor is on the x-axis with a line for each region. And we can see a few interesting patterns emerge here. The importance of nearshore rocky reefs in region 1. So that's showing the importance of hard bottom substrate and close to shore waters. And this fits with what we know about how gray whales feed in region 1, particularly their reliance on rocky reefs off of Newport right here. In region 2, we see the importance of areas close to or just south of major capes and near estuaries. And this fits with what we know about their feeding patterns in region 2 and these hotspots off of Cape Blanco and the mouth of the Klamath River. And then we see this some patterns across the board, like areas where that shelf is wider, and this interactive effect of upwelling and relaxation. So those two plots are showing an interaction effect with relaxation on the y-axis, cumulative upwelling on the x-axis, with the lighter colors, the lighter blue or the lighter green being a stronger impact. And so what this is showing is that basically we do see this sweet spot where there's a significant, important interaction of both upwelling and relaxation for gray whale abundance in these nearshore waters in both regions. So we can use these models then to predict abundance patterns by year over time. This little animation is flipping through some of well, it's summarizing the abundances by year in these coastal waters. So you don't have to squint too hard, and because they don't change very much anyway, we've also summarized it another way, which is showing just the mean predicted abundance by latitude across all of our study range and all of our study years. And we again see this emergence of these long-term stable hotspots and no real latitudinal shift over time. And we see that this aligns really nicely with the raw observations from the sightings data that I showed initially. All right. I'm going to skip this slide and go to this one. So here we can see with summarizing things a bit differently, and this is showing in this top plot the abundance across each of our regions per year. So this is saying basically across the whole region, how many whales were there for each year of our study, with region 1 in the green and region 2 in the blue. And you can see that these patterns these abundances sort of crisscross each other. Sometimes they align, sometimes they diverge. They don't necessarily align super well, and they don't have any real clear directional trend, which is interesting. And one sort of natural next question is, how does this relate to ocean basin scale patterns that operate on decadal scales like we have in our study here? For example, the Pacific Decadal Oscillation, or PDO, which we know regulates a lot of processes in the California current. So what you're seeing here is the relationship between gray whale abundance and the PDO in the spring leading up to the feeding season in that top panel, or the variability in the PDO in that bottom panel. And we see this interesting pattern where there's a positive relationship between gray whale abundance and the phase of the PDO in region 2. And so this was initially kind of puzzling, but led to some interesting sort of ecological musing. So bear with me here. And these ocean basin scale patterns, these differences in how these ocean basin scale patterns impact predators may be impacted or may be mediated by how they forage or what they feed on. This has been shown in a few different species of fish and seabirds, where species that feed in the water column benefit from cool ocean conditions, so negative phases of the PDO, whereas species that feed benthically, so on critters that live on the bottom, they benefit from either positive or variable phases of the PDO. And so what we think might be happening is that this is alluding to what we know about differences in what prey gray whales target between these different regions. And that the benthic prey that they seem to target in region 2 is maybe what's being enhanced by these variable PDO conditions. And so really what we're seeing here is that these differences in habitat use patterns throughout the range of these gray whales and the differences in their response to these large-scale oceanographic conditions really probably comes down to what they're feeding on and what impacts their prey. Whether it's the reef-associated feeding on mysids in the water column in region 1, or gray whales slurping up mud off the bottom to filter out these benthic invertebrates in region 2, it seems like all of these sort of habitat preferences and impacts of variable conditions come down to how they impact the prey these whales are here to eat. So to summarize this part, you can read if you can't hear me, that's fine. This is the summary part of the gray whale talk anyway. So we documented a number of important habitat features, and also that the habitat features are not, the preferred habitat preferences aren't necessarily equal throughout their range. They have these region-specific preferences. We showed that this relationship between upwelling and relaxation, where increased upwelling really seems to be beneficial if it's interspersed with these relaxation events for these nearshore predators. And there's this variation in predicted abundance from one year to the next, and that may relate to their regional foraging preferences. And so this begs a question: what are the implications of sort of changing ocean conditions for gray whales in the Northern California current? And what I'll say about that is that one of the sort of projected impacts of climate change on upwelling regions is sort of a spatial shift in the intensity of upwelling, where we may see more intense upwelling conditions further north. And so I'll just pull up this interaction plot for region 1 again, and we documented this sweet spot where it's not just upwelling, but there's relaxation events that are really important here too. And so if we're getting more intense upwelling with less frequent relaxation, we may be headed to a sort of less favorable set of conditions in this region. Okay, part 2. So in the case of harbor porpoises, we were interested in examining whether harbor porpoise abundance and distribution patterns reflect their habitat preferences and also the availability of their prey. And we hypothesized here that their prey association patterns differ regionally throughout our study range, and that their fluctuations in abundance or the population size might be related to patterns in overlap with their different prey, or the availability of their prey. So before we go further, let's zoom way out and think about some sort of big picture ecology here, which is just, let's pose the question: how do predators, mobile predators, respond to shifting prey availability? And we'll put out just a few different scenarios here that we're going to test. In one case, they can shift their distribution in response to changing conditions and prey availability. So in sort of a null scenario, abundance fluctuates similarly across different regions, different areas. You'd see a positive relationship between abundance in one place and abundance in another place. But if there's a distribution shift, we might expect to see an inverse relationship, where as abundance increases one place, it decreases somewhere else due to movement from one place to another. In another scenario, we could look at the overlap between predators and prey and see whether predators are potentially switching prey in response to changing conditions. So here, those squiggly lines represent overlap with different prey types. And we could imagine that in a null scenario, the overlap is pretty similar between the predator and different prey species, whereas in an alternative scenario, there's again an inverse relationship, whereas overlap with one prey type is going up, overlap with another prey type is going down, indicating a switch. But let's think about a third scenario where the abundance of a predator depends on the overlap with a specific prey species. So here in a null scenario, we have overlap with two different prey types, and the abundance is sort of independent of overlap with those prey. Whereas alternatively, we could see a relationship where the abundance of a predator shows a sort of a directional relationship or depends on overlap with their target prey. Okay, let's reframe this for the purpose of our study. Sorry, I had to. All right. So in this case, we're interested in understanding how do harbor porpoises respond to shifting forage species availability in our study area. We did this by first making, again, these density surface models to predict distribution and abundance. We then looked at the overlap between predicted harbor porpoise distribution and the distribution of potential prey species, and then related abundance to overlap. So again, we made these models by region based on these sort of region-specific attributes of our study area, and made these density surface models with a number of static and dynamic environmental predictors. And we got these functional response curves once again. And so here in the case of harbor porpoises, we see some interesting patterns emerge, like the consistent preference for shallow waters and soft bottom substrate. We see the sort of the importance of river estuaries, proximity to river estuaries in region 1. We see this influence of proximity to these prominent capes in regions 2 and 3 further south. And we see a sort of a variable influence of upwelling and relaxation conditions here, where in region 1, it seems like the amount of relaxation events is a bit more influential. In region 2, we see the importance of stronger upwelling, and in region 3, there was no significant impact of upwelling. So we took those models to make our spatial predictions, took those to get our abundance estimates by year and by region. And so we come up with essentially these annual predicted abundance estimates within each of our region. For harbor porpoise abundance in each region. And so remember here, we wanted to, for scenario 1, seeing if they switch their distribution, we wanted to see whether these abundances fluctuate together or differently, right? Whether there's a positive relationship or a negative relationship or no relationship. So here we can see that there's generally a positive relationship in the abundance between regions 1 and 2 and between regions 2 and 3. Not much of a relationship between regions 1 and 3, which are farther apart from each other. But in no scenario do we see a negative, or in none of these examples do we see a negative relationship that would indicate large-scale distribution shifts between years and regions. So to move on to the other scenarios about prey switching or relationship with prey overlap and abundance, we needed to look at overlap with different prey types, which begs the question, what the heck do harbor porpoises eat? And so as you can see here, this is like a pretty typical look you might get at a harbor porpoise. In fact, this is a really good look at harbor porpoises, where they're diving repeatedly in one area, indicating they might be feeding. And even if you get good looks like this, it's very difficult to observe any of them actually eating anything. However, stomachs of stranded or bicaught porpoises have contained a number of different forage species in various locations throughout our study area, including herring, anchovy, sardine, market squid, and sometimes smelt and hake, sometimes a smattering of other things. Fortunately for us, data on the occurrence of several of these different key forage species are available throughout our study area. So we wanted to measure the overlap between the distribution of of harbor porpoises. We got this from our density surface models that we looked at, and measure the overlap between those spatial distributions and the availability of different forage species. And specifically, we looked at anchovy, herring, sardine, and market squid. And we picked these for one because they are have showed up in harbor porpoise stomachs, so we have good reason to believe that porpoises eat them. And secondly, because there are some amazing available data products championed by Barb Muelling and her colleagues, who have taken really a whole lot of trawl survey data, related that to regional ocean modeling data to produce these really lovely daily resolution spatial layers of the probability of occurrence of these different species. They've also very generously made these data sets available on ERDAP, so I'll highlight that resource here and say thanks again to Barb Muelling for all of her efforts here. So we compared these two sets of distributions with what's called a Hurlburt's index of spatial overlap. It measures basically the encounter rate between predator and prey. There's the math if you want it, but basically it takes the proportion of predator and prey that are in a given location and accounts for the area occupied versus the area available. And so essentially what you're doing is comparing two different spatial distributions, how well they align, how correlated they are. And I'll also just flag here that this overlap analysis was motivated by a lovely review study led by Gemma Carroll that does a great job of comparing different ways of assessing predator-prey overlap patterns. Okay, I am not going to pretend to be an expert on the biology of any of these forage species. In fact, I think there's a lot of expertise in the room and here on campus in these species. But I am a spatial ecologist, so we can summarize things spatially and look at their distribution relative to porpoises. So what these colorful heat maps are showing is on the y-axis is the day of the season that we're interested in. So that's between mid-May and mid-August. On the x-axis is the year, and so this is basically, and then we're looking at this by region for each species. So this is just a way to summarize the availability of these different potential prey species. And some patterns pop out, like these dramatic fluctuations in the availability of anchovy, how herring are really available in region 1, that seems to be where they're going to be playing a role. Market squid, we see this increasing availability across regions. And sardines, we see sort of fluctuating availability and a decline over time. So now let's look at the overlap patterns between harbor porpoises and the occurrence of each of these species compared to each other to test scenario 2, which is evaluating whether these predators might be switching what prey they're targeting. So each of these dots is the Hurlburt's index of overlap between harbor porpoise and anchovy on the y-axis and between harbor porpoise and sardine on the x-axis. And we're generally seeing a positive relationship in these overlap patterns between the different prey types. And so this doesn't indicate that prey switching hypothesis, in which case we would see a negative relationship, right? If they're overlapping more strongly with one and less strongly with another over time. And this relationship, I've showed it just for anchovy and sardine here, but this relationship was generally consistent across different study species and across our study regions. All right, so no strong evidence for prey switching. Let's take a look at scenario 3, which is a bit nuanced, but this is that we're testing whether the abundance of harbor porpoises is related to overlap with any of these particular prey species. So here's a lot of lines going in different directions, but let's walk through it. What this is showing, so here on the y-axis, we have the predicted abundance of harbor porpoises. On the x-axis is that index of overlap. And each of the colors is for a different potential prey species. And what's interesting here is that the lines go in different directions for different regions. So we're seeing region-specific relationships between harbor porpoise abundance and overlap with these different prey species. So we see a positive relationship with herring in region 1. I should say that herring probability of presence was so low in the other two regions that we only looked at it for region 1. So we see this positive relationship between abundance of harbor porpoises and overlap with herring in region 1. In region 2, we see a positive relationship with anchovy. And in region 3, we see that positive relationship with market squid and sardine. So this is interesting because it does indicate that they may be responding, or their abundance may be related to overlap with preferred prey or target prey in different regions. So let's go back to this question about how they respond to shifting prey availability in our study region. We don't see evidence for any large-scale distribution shifts between regions over time. We don't really see evidence for shifting, for switching prey, at least based on their overlap patterns. I will say there might be one slight exception here, which is sort of a weak relationship that may indicate some shift between anchovy and market squid, with the increasing availability of market squid in region 3. And then we have scenario 3, where we found that we did find that these abundance of these predators, the abundance of harbor porpoises, was strongly correlated with overlap with region-specific prey. All right. So let's summarize this part, looking at harbor porpoise distribution and abundance and prey overlap in our study region over the last 3 decades. We found that they didn't shift their distribution over time, and that their distribution was related to a number of different preferred habitat characteristics, including shallow, soft bottom substrate habitat, and in certain locations, proximity to things like river estuaries and prominent capes. We found that harbor porpoises did not seem to be switching prey over time, with this possible exception relating to increasing market squid availability. And there is an obvious caveat here, which is that these are predicted distributions. This does not equate to actual consumed prey of the harbor porpoises eating these fish. But we do have reason to believe that they eat these forage species, and we do see patterns in their response to the availability of these prey species. And finally, we did find here that their abundance is related to overlap with specific prey species, with key regional differences in those preferences. So sort of in a big picture or forward-thinking take on here at the end is that the next question is, how will these species, these are sensitive nearshore predators, they're small-bodied in cold water, and they need to eat all the time. And so sort of this framework for how are they responding to these variable conditions, variable prey availability, can hopefully help shape how we think about managing for or conserving the persistence of these populations of these nearshore predators and the ecosystem that they rely on. All right, so I'm going to wrap it up by repeating the title of this talk and emphasizing this part, which is 3 decades of nearshore surveys. And so I had the real privilege of sort of inheriting this data set in its 30th year. I had the pleasure of joining the field team on the water a handful of times, both when it looked like this and when it very much did not. And so I want to acknowledge the sort of forward-looking protocol development, really, by the folks that started these surveys in the early 90s to record all of these different species, the decades worth of field teams that collected the data, and the generosity of Craig Strong and of these folks to share the data with us so that we could get at these research questions that we wouldn't be able to answer otherwise. And I want to acknowledge the many folks that made this work possible. So Lee Torres, my advisor, she caught wind of this data set at a PCFG consortium meeting and mentioned it at lunch. And then we started chatting and dreaming up this study. So yeah, really fortunate to have Lee's distribution modeling wisdom along the way as well. Craig Strong championed the data collection along with the many folks from Crescent Coastal Research And Barb Mueling is a distribution modeler extraordinaire and contributor of these amazing forage species layers. And many folks in the room and otherwise have contributed valuable insights into the ecology and the technical computational aspects of this work as well. And I also want to thank all of you who are Oregon gray whale license plate owners, because the dollars from the Oregon gray whale license plate sales have directly supported this research on Oregon marine mammals. And with that, I'm happy to take questions in the time left, and thank you for listening.
Great. Thank you so very much. So we're going to open up for questions. We'll start with folks online. Roseanne, do we have a question online yet? Okay. So for folks online, if you put any questions into the chat, we'll work through those. How about questions in the room? Okay. I was going to say, you cannot let Dawn off.
Great talk, Dawn. This is my marine mammal naivete, but I'm assuming you can only see them when they need to go to the surface to breathe, or if there's prey at the surface. And so I guess my question is, how do you account for potential biases across species based off of the fact that you're only going to see them if they're breathing and not doing other things, maybe beneath the boat for extended periods of time?
Yeah, great question. That is part of why this density surface modeling approach is powerful, is because of our ability to account for things like that, at least to the best that we can. So one way to account for, I mean, you're absolutely right. These animals, you can only see them when they're at the surface. And so one thing is a standardized survey speed. So the boat goes at a speed that is basically meant to ideally not overtake and miss animals, but also not go so slow to where you're double counting. There's this step called accounting for availability bias, and that basically is a way to include an estimate of the proportion of time that you are able to detect an animal at the surface. And then there's the observation conditions that get included as covariates in the detection function. So that's things that are going to limit your availability to see an animal when it's at the surface. So that's things like the Beaufort sea state and other descriptions of the observation conditions at the time. So that all goes into this detection function about your probability of detecting anything, which happens before we even get to the distribution modeling phase. Yeah, good question.
All right. Looks like we have a question online. Go ahead, Roseanne.
Is the high abundance off Newport in any way related to the ability to mobilize quickly during good weather windows? And nice talk. Nice job.
Thanks. That is a great question and a very fair question. Oftentimes, sightings are biased by where people are looking. In this case, the field team has an objective to cover the entire region, like the entire, or at least part of it, in a field season. And so they generally really do. So with this data set in particular, I feel pretty comfortable saying it's not super biased by proximity to, they'll camp in a certain area to be closer to a boat launch for a good weather window, for example. So they really do try and have even coverage. And the other part I'll say is that we do account for survey effort in our analysis as well to try and mitigate that impact if it is there.
Great. Thank you. Questions in the room?
Thank you, Dawn. That was super interesting and really nice to see harbor porpoises get some attention. I feel like that's
right.
They're
yeah.
everywhere and yet no one studies them. I do have a gray whale question though. So this could be naive. as someone who doesn't know so much about the PCFG, but one of the things that stood out to me in one of your early heat maps where you had abundance plotted by year and then or encounter rate by year and then also by latitude, it looks like the highest abundance by far was in the first year of the survey, like in 92. And I was just wondering if there's been a general decline in gray whale abundance off the coast and if you know why that's happened.
That's a great question, one I might defer to some of the more PCFG experts in the room, but I know what you're referring to, that high peak in it's in encounter rate. It doesn't come through quite as strongly in the predicted abundance, but there is a high peak there early on. One thing that is kind of interesting, and I don't really know how much to read into this, is in our encounter rate data, there do seem to be peaks in encounter rates right before unusual mortality events. And in terms of whether PCFG abundance has stayed stable, that's something I don't know off the top of my head, but I think it has been pretty stable. I think there's been larger fluctuations in the greater like ENP stock, but that PCFG gray whale abundance fluctuates a bit due to some recruitment, whether internal or external, but generally fairly stable. How'd I do? I'm getting nods. Great.
I was going to crowdsource it, but we just in the room got hands and thumbs up. So for folks online, that's the result we got. Okay. We have any other questions online, Roseanne?
Yes. Was detectability of porpoises influenced by distance from shore?
Oh, that is a good question. So I didn't look at whether the probability of detecting, like your ability to make a detection related to distance from shore, because your probability of detecting something has more to do with the observation conditions for which we considered like sea state and stuff like that. Distance from shore was included in the distribution models where we looked at these functional relationships, and there was higher, there was a relationship where there was better porpoise habitat basically nearer to shore. But I didn't put that directly into the detection function, which is an interesting idea. I suspect that whether you can see a harbor porpoise shouldn't relate to how far from the coast you are, that has more to do with their habitat preferences.
Okay, questions in the room?
Great talk. I'm going to ask a fish question. Going back to kind of the abundance of prey species and things like that, why was the timescale mid-May to August? Did you look at either side of that going into winter as well, or is it just because summertime observations, things like that?
Great question. Probably something I should have said very clearly up front, but that's when the surveys take place. So that has to do with when they're surveying for marbled murrelets, feeding on the water. And so that is what we limited our analyses to because of data availability. Yeah.
Okay, I know there might be questions on the other side of the room, but I'm standing here, so I'm going to get this one and then I can go over the other side.
This can be a quick one because it actually was related to that timing question and what the timing of the survey was, and then how that might affect the behavior of the gray whales during migration and who might be staying off coast versus, or this coast versus heading further north, and therefore affecting the abundance estimates.
Yeah, great question. So the timing of these surveys, they technically start in mid-May. They get very few days on the water in mid-May because, well, you all who live here know how it can be trying to spend time offshore in May. And so really the bulk of the surveys are in June and July through mid-August. And that aligns really well with what is known about the time when most of the eastern North Pacific gray whales are on their Arctic foraging grounds, and the whales observed here are highly likely to be from the PCFG. So we think there's minimal impact of ENP gray whales being observed, biasing the abundance estimates. That being said, animals can certainly move around within a year and between regions. And so that is part of why we made the abundance estimates come from daily predictions that were then averaged for a feeding season for a year. So that is meant to sort of look at patterns, general patterns from one year to the next without having a huge bias based on animals moving in or out of areas within a season. That being said, we did look at abundance estimates within a season, and there's variability from one year to the next, but we don't see like a hugely dramatic like peak or decline or something during the periods that we're predicting to.
Okay, I
Hopefully that made
apologize
a bit of
again. Blame
sense.
Scott here. We've got questions around the room, but he.
Great talk, Dawn. So I'm stretching to try to figure out kind of like the feeding ecology that's happening for the gray whales in the different regions. And this is a terrible question for a quantitative ecologist, but can you like turn that into a cause and effect for me? In other words, do the different attributes of the habitats up and down the coast drive prey availability that result in whales feeding in different ways and having different distributions because of the provision of resources that provide those prey? Or is it you have whales that feed in different places and you're seeing that in your data up and down the coast? What you get what I'm getting at? Like, what's the cause and effect in the geographic pattern that you see?
That's a great question. It's a good sort of chicken and egg thought experiment, Scott. I think, well, the short answer is I don't know, and I don't think anyone knows. Having said that, my inclination is to lean toward your option one. And so gray whales are really flexible foragers. And we even, I mean, folks in this room are studying aspects that like even individuals have certain preferences, and within different regions, there seems to be habitat preferences. So I guess my inclination would be to lean toward your option one, where these different environmental characteristics give rise to favorable conditions for different prey items that are worthwhile for gray whales to feed on, and they target those in these different regions. We can muse about that more though. Okay.
Dawn, thanks. Great talk. It's great to see the analysis of this amazing data set. I just have a quick thought for you as you write this up. As you may know, there's been a tremendous amount of research done on the molecular genetics and distinction of harbor porpoise along the U.S. West Coast. And that has resulted in the designation of many harbor porpoise stocks, actually that go all the way up into Alaska. It's been very frustrating for the regulatory side of NOAA, because the more stocks and the smaller the spatial distribution of them, the more complicated the regulation becomes. But until I saw your results, I've never seen any significant analysis to address how those great many stocks may have evolved. And I think your results with this amazing data set over not only such a long period of time, but a great spatial extent might actually be able to touch on that. So just for your consideration in the discussion of your paper.
Thanks, Lisa. Yeah, that comment is very timely because as I've just written the discussion of this paper, which is now with co-authors, the scientific review group for West Coast stocks of marine mammals is happening right now, and they're actually reviewing, and it's likely to move forward, designating a Central Oregon stock and revising these stock boundaries. And I have a whole sort of ongoing discussion of, well, do we actually redo these with stock boundaries instead of these biogeographic boundaries? So that's probably a next step that's going to happen, is to look at it by these stock boundaries. But what's cool to me is that it seems initially it was like, do we go with like biogeography, or do we go with lines drawn for management? And it seems like with these, and my inclination was, let's go with the biogeography. We're taking a habitat-based perspective here. However, it seems like the two are actually on a, I was going to say collision course, but I think it's positive. I don't know. And so, like the recent genetic evidence does find, I mean, it finds this isolation by distance throughout their range, but these sort of greater than expected differences in genetic population structure around biogeographic boundaries like Cape Blanco. And so it is cool that the sort of habitat, or what we know about the ecology of this region, is reflected in the population structure throughout their range. So yeah, thanks.
All right, we're
We'll
gonna take
keep
one
working
more
on it.
question in the room and we'll let Don off the hook.
I heard that there was someone sitting on the other side of the room. It must be me. I'm the only one on this half of the whole room. Great talk, Don. Thank you very much. Amazing data set and great, very interesting analyses. Following up on what Scott's question was and your discussion of it, did you guys actually get prey density data for the areas that do those data exist? I'm guessing they probably don't.
Yeah, great question. Totally fair question. The answer is I wish. And so there are prey data available in sort of pretty specific pockets. So here in Newport, in the last 5 or so years, for example, there's also, and it really, that sort of in situ sampling takes like really dedicated field effort, whereas on these nearshore, and the nearshore is an area that's hard to work in, and so on these surveys they really are just driving along a line and recording observations. So our musings about what the gray whales are eating in different places do come from these sort of pockets of studies off Newport, off Port Orford, off the in sort of Humboldt, Mendocino. But it does align with observations of how they feed in different places. So at the more southern end, folks have done some work looking at what benthic prey is available and what is or was there in places where gray whales are foraging. And likewise, in our region 1, either light trap or net sampling for what sort of prey are available in the reefy areas where the gray whales feed here. But a region-wide systematic prey sampling effort would be beautiful. Yeah.
All right, thanks. I've got one more question if you'll take it still.
Sure. What you got?
With Pacific sardine being one of the preferred prey of the harbor porpoise in your southern or region 3, as I recall, which is south of Cape Mendocino. You may be aware that the northern stock of sardine are no longer going south of Cape Mendocino and have not been going south of Cape Mendocino since about, I believe it's 2014. And I'm just curious your thoughts on that and how it reflects, you know, how it's related to what you're seeing and what your results are.
That is a great question and something I think I'd love to chat with you about more. Like I said, I'm certainly not an expert in the ecology or biology of these species. And that would be a good question to pose to Barb about how that shift in the distribution of the different stocks of sardine, how well that is reflected in these predictions of availability. over time. So that's a great question. I don't have any real answers, but definitely something to consider. Thanks, Greg.
All right. Thank you, everyone. Let's give one more round of applause for our speakers.
Thanks, Cinnamon.
And Don, thanks so much for letting us be your guinea pig while you present this information to us. I appreciate it.
Thanks for
All
listening.
right, everybody. We'll see you next week. Thanks for being here. Thanks, Post Online. I'll see you next week.